Hi, my name is Vidushi Agrawal, and today I will be discussing natural compounds in cancer therapy. Cancer is the second most common leading cause of death in the world. There's no denying the importance of cancer research. Natural compounds play a crucial role in developing new cancer treatments. To understand how these natural compounds work, it's first important to take a look at some of the main current cancer therapies today. Major treatments for cancer include using immunotherapies and DNA-damaging agents. Immunotherapy fights cancer by boosting or changing the immune system, and there are 5 primary methods of it. Clinical trials are ongoing to test their effectiveness in various cancers. DNA-damaging agents like chemotherapy are widely used and work by damaging the cell's nucleus, therefore disrupting cell division. DNA-damaging agents and immunotherapies both trigger apoptosis, which is a natural mechanism for programmed cell death. A wide variety of conditions in the body can lead to the activation of the apoptosis pathway, notably DNA damage or uncontrolled proliferation. Apoptosis-inducing drugs are the most successful and effective non-surgical cancer therapies and work for all types of cancer. Identifying and studying medicinal plants can lead to the discovery of novel bioactive compounds with potential anti-cancer properties. So why does this matter? While significant progress has been made in cancer treatments, it is important to acknowledge that they have not provided a complete and universal solution and that challenges are still present. Ongoing research and advancements are essential to address these problems and enhance the efficiency of cancer therapies. In other words, research on medicinal plants is critical to advancing cancer treatment. The natural compounds I'm going to cover in this presentation are camptothecin from the Camptotheca acuminata plant, andrographolide from the Andrographis paniculata, and beta-glucans from the Agaricus blazei mushroom. Camptotheca acuminata, also called the Chinese happy tree, is a deciduous tree native to South China. In the 1960s, researchers identified it as a potential anti-cancer agent due to its unique alkaloids, especially camptothecin, which showed promising cancer-fighting properties. CPT and its derivatives are powerful inhibitors of an enzyme called DNA top 1, which is crucial for DNA processes like replication and repair. DNA top 1 works by unwinding DNA strands so they can be copied or repaired and then sealing them back together. CPT and its derivatives stop this process by binding to the enzyme DNA complex, preventing the DNA from being resealed, which leads to strand breaks and eventually causes cell death. Researchers have developed derivatives of CPT such as irinotecan and topotecan, which are better tolerated by patients and are valuable in cancer treatment. Irinotecan is used to treat cancers like metastatic colorectal, lung, and pancreatic cancer, while topotecan is mainly used for ovarian and small cell lung cancer. Side effects include mild to moderate nausea for irinotecan and bleeding gums, darkened urine, and darkened stool for topotecan. Ongoing clinical trials are continuing to explore and document these side effects. Andrographis paniculata is a medicinal herb native to India, Vietnam, and Sri Lanka, widely used in traditional Asian medicine for treating stomach aches, inflammation, poisonous stings, and more. Its active compound, andrographolide, is primarily found in the leaves, with smaller amounts found in the stems, roots, and flowers. Andrographolide inhibits cell proliferation by preventing cell cycle progression and inducing apoptosis in cancer cells, which is its primary mechanism of action. It induces apoptosis in tumor cells, inhibits cell proliferation, and blocks the cell cycle, which leads to cancer cell death. Additionally, it impedes metastasis by blocking the bond of cancer cells to the blood vessel lining endothelial cells, inhibiting the formation of new vessels. Andrographolide enhances the immune system function by stimulating the production of white blood cells, the release of interferon, and activity of the lymphatic system. Andrographolide has demonstrated its efficacy by targeting specific molecular pathways involved in developing cancers. These include breast, lung, liver, colon, renal, and cervical cancers, as well as leukemia. In clinical use, dosing methods of andrographolide and its derivatives consist of compounded tablets, intravenous injections, and sitz baths. Due to its immune-stimulating and anti-cancer properties, Andrographis paniculata is a potential candidate for preventing and treating various cancers and tumors. Agaricus blazei muriel is an edible mushroom first discovered in 1944, commonly used in teas and cooking. By the 1980s, it gained attention for its medical and biochemical properties, showing potential in various health applications. The anti-tumor effects of ABM are thought to be connected to how it interacts with different cytokine networks, which help regulate both the innate and adaptive immune systems. Beta-glucans are some of the most involved active molecules in the fungus, which are believed to stimulate the immune system. They stimulate immune cells like macrophages, NK cells, and dendritic cells, boosting immune response against cancer. By enhancing NK cells' ability to directly target and kill cancer cells, beta-glucans help to inhibit tumor growth and improve cancer cell elimination. They also directly block cell growth, arrest the cell cycle, increase apoptosis, and affect key signaling pathways related to tumor progression. They influence various signaling molecules, which also leads to cell cycle arrest in specific phases and promotes cancer cell death. Aragol is one of the most extensively tested and widely used derivative of ABM. Aragol-induced apoptosis is associated with reduced mitochondria membrane potential and release of apoptosis-inducing factor from the mitochondria to the cystosol. It upregulates BAX, which promotes programmed cell death, and downregulates BCL2, which typically promotes cell survival. Agaricus blazei has been studied concerning various types of cancer, including prostate, lung, colorectal cancers, leukemia, and for multiple myeloma, a phase 2 trial is underway. Considerable work has been done in the preclinical phase, involving laboratory studies with primarily cell cultures and animal models. ABM is primarily consumed as a part of the diet with the addition of supplements. The anti-tumor properties of Agaricus blazei shed light on its potential as a valuable supplement in cancer therapy. So, what is the future of these natural compounds? Future research should focus on several areas to fully realize the potential of natural compounds in cancer therapy. For camptothecin and its derivatives, efforts should aim at developing formulations that minimize toxicity while maintaining efficacy. The most common way of intake of CPG and its derivatives is orally, but a novel delivery method is in development: nanotechnology. Nanotechnology offers advantages such as targeted delivery, reduced systemic toxicity, and improved imaging. For andrographolide, more extensive clinical trials are necessary to confirm its effectiveness and safety across different cancer types. Combining andrographolide with existing therapies could also be explored to enhance its anti-cancer effects. Additionally, more extensive clinical studies on ABM are needed to validate its efficiency and safety as a cancer therapeutic. Similar to andrographolide, investigating its potential with other anti-cancer compounds could offer new insights into the cancer treatment. In conclusion, natural compounds such as camptothecin, andrographolide, and beta-glucans offer promising avenues for cancer therapy. With further research and clinical validation, these natural compounds have the potential to significantly impact cancer treatment and improve overall patient outcomes. Thank you to my wonderful mentor, Matthew, for his continuous encouragement and support, and the entire Polygence team for providing me with this amazing opportunity. Thank you for listening.
Thank you so much, Vidushi. So what initially interested you in this topic?
So I've always had a sort of passion for biology, and there wasn't really a specific moment where I was like, oh, I'm really interested in cancer. I did in Science Olympiad, one of the events I did was micromission, which is related to that, and I really found that event very interesting. So since then, I've just been trying to do as much as I can to just kind of gain more knowledge on this topic.
That's great. And where would you like to go next with this?
I would like to first explore more natural compounds. Like these are just three of many. Um, Also, it would be amazing if I could take my review paper to a lab and test out some of these natural compounds for myself, testing them in a proper lab setting.